Okay, right now we're going to be reviewing Chapter 48, which is Neurons, Synapses, and Signaling. And you can follow along with the review sheet that we printed out and passed out. So a basic intro to the nervous system. Neurons are the cells that make up both the central and the peripheral nervous system. The central nervous system, the brain and the spine basically make it up. And it's full of interneurons, which are going to be the cells that process the signals that it's going to receive from the sensory neurons that are in the peripheral nervous system. And they're also going to be the ones passing on messages to the motor neurons, which are also in the peripheral nervous system. And this entire process, we're going to go in depth with it, and it's basically how you receive and transmit signals throughout the body.
Alright, so here's a diagram of your basic neuron. You have your cell body, which is the weird part. It looks like it has lots of hairs. And then probably the most important part, arguably, is the axon, which is the part with all the pink lumps on it. And this particular axon is myelinated, and those are the pink lumps. And basically a myelinated axon is a axon that has coding on it. And the coding allows for a signal, after it's transmitted from the cell body, to jump over these little gaps. To jump over these little gaps, and it basically speeds up the signaling process. It jumps over the gaps, and they're called nodes of Ranvier. And in a myelinated axon, it's the conduction is called saltatory conduction. And basically, overall, in any transmission of a signal, you have the signal traveling down the axon, and it reaches the axon terminals where it gets passed on to either a muscle or another nerve cell.
Okay, so right now we have a quiz that we were going to do in class and pass out cookies for correct answers, but since that's not a possibility, right now we're just going to use it as a review. So the first question is, why is a myelinated sheath better for conducting signals, and what types of glia produce myelin sheaths and where? The answer to that question is the myelinated sheath is better for conducting signal because, just as Tanner described, it's going to cause the electrical signal that's moving down to move a lot faster so it can pass on quickly. And the two types of glia that produce myelinated sheath are oligodendrocytes and Schwann cells. Schwann cells produce myelinated sheath in the peripheral nervous system, and oligodendrocytes produce myelinated sheath in the central nervous system. What part of the neuron receives signals and where are they passed on? Just as Tanner was saying, with the hair-like structures that are coming off of the cell body, those are dendrites, and they're going to be what's receiving the cell signal that's moving on, and it's going to be passed on through the axon and out of the synaptic terminals. The optic nerve is what type of nerve, and in which nervous system is it found? The optic nerve is a sensory nerve, and it's found towards the back of your eye, and that makes it a peripheral part of the nervous system because it's going to be moving like a stimuli to the interneurons, which is then going to cause a motor response.
Alright, so how is a signal transmitted? Alright, the most important part of any signal is action potential. So what is action potential? Action potential is the level of membrane potential that a cell must reach before a cell response is triggered. So how is it created, and what key factors in the membrane help to facilitate the transmission of a signal? Essentially, you have this process called depolarization. And depolarization is where sodium and potassium channels open, and you get a ton of sodium ions flooding in and a ton of potassium ions floating out. And this causes the membrane to be extremely polarized. And it, as you can see by the diagrams, it begins to polarize the neighboring sections of the membrane, and so it's able to travel all along the axon. And after this happens, you have the repolarization, as you can see also by the diagram, indicated in green, and that's after an action potential is reached, and that's making the cell return to a normal state.
Okay, so the basic components of the action potential, we've got voltage-gated ion channels, and those include the sodium and potassium ion channels, and you've also got the sodium-potassium pump. Now, specifically, voltage-gated ion channels are exactly what they sound like. They're ion channels that open up in a response to a stimulus, which in this case would be the change in the membrane potential. And this change has to reach threshold, which is a certain level of depolarization before before an action potential can happen, because it's an all or nothing reaction. These potassium and sodium ion channels are what move in a volley of opening and closing down the axon. And the sodium channels are going to open first. Sodium's going to flow into the membrane, and in response, they're going to close as the potassium channels open behind them, and potassium's going to flow out. So now you've got a complete flip of the concentration gradient along the membrane. And once that's finished moving all the way down the axon, the sodium-potassium pump is going to fix the concentration gradient by moving the ions back to where they should be.
Alright, so here's another quiz. So what is the exact formula for calculating equilibrium potential of a cell? The exact formula is 62 millivolts times the log of the ion on the outside over the log of the ion on the inside. And that would be worth 2 points. What are all stages of the membrane must move through when transmitting a signal? Well, you have the state originally at resting potential, and then it must reach threshold for depolarization to start, and then you have depolarization, and then you have the action potential. Then you have the repolarization, and then the undershoot, which is the state where the membrane potential drops below resting potential, and that's where the sodium-potassium pumps come in to re-regulate it.